Today's class, we'll look at some of the biomolecules. Basically, we'll be looking at the amino acids, which are considered as the building blocks of proteins. And in turn, the proteins are the building blocks of the organism. So that's one easy way we can define amino acids. They are the building blocks. And these building blocks we see in case of proteins, polypeptides, and proteins will form the building blocks of the organism. A second way we can define amino acids is amino acids are monomeric units. That is basic units, monomeric units of proteins, polypeptides, and peptides. So in all these cases, one amino acid will be linked to the adjacent amino acid through a peptide bond or an amide bond. So one amino acid gives its alpha carboxylate group, the other amino acid gives its amino group. This is what we call it as a peptide or amide bond. So this corresponds to amino acid 1, this corresponds to amino acid 2. Defining the amino acids is based on their functional groups. So when you look at an amino acid, you have at least 2 functional groups. So these 2 functional groups we find in every amino acid. Other amino acids may have additional functional groups. The 2 functional groups are an amino group and a carboxylic group. So these 2 groups we find in all the amino acids. In addition to these groups, we may find other groups. For example, when you take amino acids such as serine 309, you find the presence of hydroxyl group. When you consider aromatic amino acid, you find an aromatic group. If you consider cysteine, you have a thiol group or sulfhydryl group. If you consider methionine, you have a thioether group. If you consider asparagine and glutamine, you have an amide group. So amide, thiol, thioether, aromatic hydroxy. So what essentially it means is amino acids can have multiple functional groups, but every amino acid contains two invariable groups from which it is getting the name amino acid. One group is the amino group, second group is the carboxylic group. If you take the normal amino acids, naturally occurring amino acids, the amino group is attached to the alpha carbon. So if you go back to the nomenclatures, the alpha carbon will be the adjacent carbon to the functional group or to the priority functional group. So here the priority functional group is carboxylic group. So we'll write this as carboxylate group. This carbon is what we call it as alpha carbon. This alpha carbon has amino group. So this is alpha amino acid. It has alpha hydrogen and then there is a side chain group. So those are the four components that we typically find when you look into the structure of an amino acid. Alpha carboxylic group, alpha carbon, amino group, and then the R group. For most of the amino acids, or almost all the amino acids, this will remain the same. This will not be changed. So this is common for all the amino acids. The only difference is proline, where you don't get an amino group, instead of that you get an imino group. So since these groups are same for all the amino acids, this region of the amino acid is responsible for common properties of amino acids. So gives common properties to amino acids. So what gives the common property is the common group that is associated with the amino acid. What gives a unique property is the side chain, the R group, which is unique, which distinguishes one amino acid from another amino acid. So this is distinct for each amino acid. So it is responsible for individual, distinct properties of amino acids. So that's how we can divide the groups which are present in the amino acids into two distinct categories. The first one, the general groups which are common for all the amino acids. They give common properties to the amino acids. And the second one, the unique group, the side chain group, which is distinct for any given amino acid, and it is responsible for the distinct properties of each amino acid. This another way we can discuss the amino acids is these are amphoteric compounds. So here we are extending the first definition. First definition we said these are bifunctional compounds having an acidic group and a basic group. Now the acidic and basic groups we can extend into an amphoteric nature. An amphoteric compound is something that acts as acid as well as base. So a simple amphoteric compound is water. Water can dissociate giving proton plus OH minus, proton, the acidic one, OH minus, the basic one. For defining the acid, we use the definition, an acid is a proton donor. So it releases proton. Whereas a base is a proton acceptor. So it accepts proton. So how you identify the amino and the carboxylic groups present in the amino acids as either acidic groups or the basic groups. So one group that we have is COOH group. This we will see when the pH is low. This group undergoes dissociation, forming COO minus, releasing proton. The second group that we can find is the NH3 plus group. It can also undergo the dissociation and can give rise to the amino group, which is NH2 here, plus releasing the proton. So these two represent acidic groups. They are proton donors. These two groups represent basic groups. That's because they are proton acceptors. So this is how we take the second definition. Amino acids are amphoteric compounds having both acidic and basic groups. The acidic group is one which donates the proton. The basic group is one which accepts the proton. And when you look at the molecule, the acidic groups are the COOH group, the NH3 plus group. When you take the basic groups, you have COO minus group and NH2 group. Now at any given time, any given condition, whether a group exists in the acid form or a group exists in the basic form depends on the pH of the solution. So whether you can say amino group as acidic group or a basic group depends on the pH. Similarly, whether the COOH group will remain as COOH group and acts as acidic group, or it goes to COO minus and acts as a basic group, again depends on the pH. So when you have low pH, that's close to 1, carboxylic group will be COOH, amino group will be NH3 plus, both are acidic. When you have high pH, carboxylic group will be COO minus, amino group will be NH2, so both are basic. At physiological pH, amino group will be NH3 plus, carboxylic group will be COO minus. That means this is basic group, proton acceptor, this is acidic group, proton donor. So this is something that you have to remember. Based on the pH, how you can identify whether the given group is acidic or basic. The next feature is asymmetric in amino acids. And anything that has asymmetry usually has optical activity. An exceptional amino acid is glycine. Glycine has no asymmetric carbon. Because it has no asymmetric carbon, it is optically inactive. So that's why whenever you say about glycine, you simply say it as glycine. You don't say D-glycine, L-glycine. Glycine also has no beta carbon. That's another feature which is important for glycine. That's why it often acts as a hinge amino acid. Wherever protein has to undergo bending, the side chain of glycine, which is hydrogen atom, will allow that bending. The rest of the amino acids contain asymmetric center. Now when you look at the asymmetric center, you have a carbon whose tetravalencies are satisfied by 4 different groups or 4 different atoms. So if A is not equal to B, not equal to D, not equal to E, then this carbon will become asymmetric. Or chiral carbon. So this is the tetrahedral structure of your central carbon. Once you have the asymmetric carbon, this will be optically active. If it is optically active, it means it rotates plane polarized light. Since in the case of the amino acids, typically we find the L amino acids, in some of the solutions they will have levo rotation. So if I take an example of an amino acid which contains the asymmetric carbon, then I can take alanine. So if you take in terms of earlier case, A is COO minus, B is NH3 plus, C is CH3, D is hydrogen. None of the four groups are similar, so this is chiral carbon. Or asymmetric carbon. Now here we have to decide which one is the priority group. For priority group, we take the atomic mass. So it is hydrogen least atomic mass or least atomic number. Its atomic number is only 1. Here also we have hydrogen, but there is nitrogen. Nitrogen atomic number is 7, atomic mass is 40. So that means this group will become priority group. This priority group is located on the left side. So this will be called as L-alanine. Now for this particular compound, we can get a mirror image. So again, asymmetric carbon, carboxylic group, hydrogen atom. So this will become now amino group, hydrogen atom, side chain group. So priority group is on the right side. Since the priority group is on the right side, we call this as D-alanine. So if you look carefully, the L-alanine and the D-alanine, they are just like your hands, chirals, that cannot be superimposed. So if you try to superimpose them, your thumb finger will superimpose with your little finger will superimpose with your thumb finger. This kind of mirror images that cannot be superimposed will be called enantiomers or enantiomorphs. Most of the naturally occurring amino acids, those that will be occurring in the proteins, they are L-amino acids. So L-amino acids typically occur in proteins. Whereas D-amino acids occur in peptidoglycan, which is bacterial cell wall, some antibiotics, so all our 20 standard amino acids are that type. The next one that we will be looking at in the amino acids is the standard and non-standard amino acids. A standard amino acid is one which is coded by the genetic code and has its own codon. So it is genetically coded, has one or more codons, and it is directly incorporated during translation. So that's how you define the standard amino acids. The standard amino acid should be specified by the genetic code. It should have at least one or sometimes more than one codons. That codons will be specific for the given amino acid. And when the translation takes place, an amino acid tRNA should carry this amino acid and it should incorporate into the growing chain of protein or peptide. We have 20 conventional standard amino acids. They are the old ones, what people have studied almost for 100 plus years. We have two newly added ones. This is about 20 years or so. So here we have one amino acid, selenocysteine, which occurs in both prokaryotes and eukaryotes. The second one, pyrrolysine, which is so far identified only in archaea. The standard amino acids have 3-letter codes and 1-letter code. So earlier we used to go for 3-letter codes, but as bioinformatics became more prominent in terms of the analysis, people started taking the single letter code as it saves the space, it saves the bits. There are about 700 amino acids which are called non-standard amino acids. They are not directly incorporated into the protein. They are not coded by the genetic code. They don't have codons. So one example is citrulline. Another example is ornithine. Another example is cysteine. Another example is iodotyrosine. Another example is hydroxyproline. So all these are non-standard. They are never directly incorporated into the protein. These are generated during the urea cycle. This is generated when two cysteines combine. This is generated when tyrosine is iodinated. This is generated when proline is subjected to hydroxylation. So they are non-standard. So at least we have 700 non-standard amino acids. Then if you look at the standard amino acids, we can give single letter code, 3-letter code, and the name. So first one, we'll start with A. 3-letter code is ALA, and the amino acid is alanine. If you take the second one, C, amino acid is cysteine. So there is no problem in both the cases because the first letter indicates the amino acid name. Third, we have D. This actually stands for acidic amino acid aspartate. So this is sulfur, this is acidic. The next letter also again acidic amino acid. Glutamate. Next, aromatic amino acid. Next one, the simplest amino acid. So this is symmetric amino acid. Next is weakly basic amino acid. So this is best buffering amino acid. Then we get branched chain amino acids. Then we get a basic amino acid. Then again we get a branched chain amino acid. And then we get sulfur containing. Then we get amide containing then we get proline. This is unique for amino acids because it is amino acid, not amino acid. Secondary amine group. So that's the only secondary amine group containing amino acids in the standard amino acids. Then we have amide group containing amino acid, glutamine. Then we have most basic amino acid, then we have an amino acid that is commonly subjected to phosphorylation. Then we have another one which is commonly subjected to phosphorylation. Then we have a branched amino acid. Then we have two aromatic amino acids. So that gives the list of all the 20 conventional standard amino acids in terms of their single code, in terms of their 3-letter code. Currently we prefer single letter codes. For exam purpose, you have to take single code which does not directly represent amino acid name. For example, N. Aspargine, you are starting with as, but its single letter code is N. Q, you are starting, it says glutamine, but its single letter code is Q. Now R, you are starting with arginine, that one. Next one, W, tryptophan. You say tryptophan, you don't pronounce that R, so tryptophan they have taken W. Tyrosine, they have taken the next letter Y. So remember single letter codes for those amino acids where the single letter code will not correspond to the first letter of the amino acid. These are the ones that can be asked in the exam.